probably going to be hearing my little dog whine. He's feeling better and he doesn't want to be contained anymore, but that's life. So we actually have a guest speaker this morning, two guest speakers, one of whom is a former 103B graduate, Tammy, and her padre, Asan. I hope I said that correctly. And they are going to be telling you about an opportunity that they'd like to offer through their club that, of course, will be a generous thing that anybody could do, but that's up to you. And they're going to be giving about a 2-minute talk about it. So are you there, Tammy? Are you able to, or Asan?
Hello. Yeah, I'm here.
There they are. There's
Okay.
Tammy. I guess we can't share both screens. So why don't you tell them about what your club wants to do?
Okay, so hello guys. So I was a prior student in Kaufman's class. I just want to wish you guys best of luck. I know you guys are in the final stretch. I just want to say that we started a club here at Ohlone called Ohlone Cards for Carriage. It's based off an international nonprofit, and we're just a chapter of it. And we're working with the international nonprofit to offer volunteer hours for people who make hand-drawn, handwritten cards. We will be sending those cards to hospitals, and I'll share my screen for just a little bit. Let me figure this out. So we have a Facebook page and we also have Instagram. We just started, so there's not much to see, but you should go follow our page. And this is an international nonprofit for those of you who are interested in checking it out. They have reached over 26 states and like several countries as well in terms of the cards. Also, if you don't have artistic talent, that is perfectly fine because there are people who aren't as artistically apt and they still are making cards. So do feel free to join us. And we're actually planning to do more beyond just cards. So we're thinking of basically starting a mentor-mentee kind of relationships where you see that in four-year colleges. I know that at community college it's kind of hard to make friends. So if you guys are interested in basically making friends here, yeah. I don't I'm having difficulty. But yeah, so if you guys are interested in basically joining us, do come on Friday. We have a meeting. It's from 4:30 to 5:30. Our advisor is Siam Sundar. So if you've taken his class before, you can reach out to him or you can reach out to us at this email. Anyways, that's it. So yeah.
Lastly speaking, if there's anyone that's interested in volunteer hours in general, because I know since this is an anatomy and physiology class, a lot of you might be pre-med majors or nursing majors, anything of the sort. The international nonprofit is granting volunteer hours for anyone that joins and helps make and submit handwritten, hand-drawn cards. And then it would look good on the resumes that you'd submit for those applications. So if you are interested, our next meeting is tomorrow, Friday the 13th, from 4:30 to 5:30 p.m. And you can email our email that I dropped in the chat box on the side if you're interested. And if you're like, oh man, this is amazing, and you want to join, that is Professor Sundar's Zoom link. You can just go ahead and save that for tomorrow afternoon.
Okay. Thank you. Thank you,
Right.
Dr.
Thank
Kaufman.
you, guys. What a nice thing to do.
Thank you very much.
All right. Have a good day. All right. The rest of you. So we were finishing up the story of how you can make dilute and concentrated urine depending on how, I'm starting slow this morning too, depending on how concentrated your body fluids are. What is the osmolarity of your body fluids? Are you trying to conserve water because you're perhaps a little dehydrated? And so today we'll start into discussing not just the kidney and how it works, but how the kidney interacts with your negative feedback loops to maintain normal blood osmolarity and fluid balance, which is the next chapter. So fluid balance and salt balance. So I'm hoping that we get through the first 4 questions of the next lecture set on fluid and water balance. But before we do that, there's kind of a challenging part of how the kidney operates, which is the urea recycling. And we could do this either way. Let me see who's here. I know Noah's here, June's here, and is Cal Sang and Jasmine here? I'm not seeing those guys. So maybe what we'll do is go ahead and let you break out again, and maybe they'll show up, and we can reconstitute that group as a group E, but they will be the ones starting because you're going to be talking about urea recycling and how ADH, the hormone ADH, acts first, and then we'll go more into hormones with the next group and how they act on the DCT and the collecting duct to allow salt and water balance to be established when you need to. So one of the themes of the kidney that I want to stress, and maybe haven't here, but in my lectures I have, is by and large, until we get to the DCT and the collecting duct, we don't regulate permeabilities to maintain water balance or salt balances. So yes, there's regulation of glomerular filtration rate that could affect those things indirectly, but hormones don't generally act on any part of the nephron until you get to the DCT and the collecting duct. Now that's a little bit of a simplification because there is the ascending thick segment will work harder when so they'll upregulate when you need to, when you're really dehydrated, and they're sensitive to that. But before this, up to this point, we have not had any hormones acting on our nephrons. So let me break you up into groups with these people who are coming in late. I'll just again pop them into the group. But I can't wait around and hope that people who are late shower-uppers are going to manage to, here I'm doing 2 things at once, and manage to slow us down. So let me name the first group, Group E, and you're going to be doing, again, like last time, 9 through 10, and that's in the lecture set question 11 on the urinary system. And I'm going to see who's here, but I'm going to be giving the same people. Jasmine is not here. June's here. Cal Sang is not here, and Noah's here. So you guys are, so far, the only 2 people in that group. The next group is going to be Group A, and you're going to be doing question sets. Questions 11, let me see, page 12 and 13. And let me make some assignments on that. I'll just put 3 of you in there so far. And then next group is C. You're doing 14 through 16. And I'll give some people that assignment. 1, 2, 3, 4 on that one. And then we'll do question C. And you're doing questions 17 through 19. And we'll assign some folks to that. So maybe that'll leave 3. So, okay, so we'll do 3 in that one. And we'll rename the last one will be question group D, questions 1 through 4, lecture 12. Okay, and here we go. I'm going to assign the last 4 of you into that group. And then we'll see as people come in. I don't have anyone else who hasn't come in yet. All right, let's give you 15 minutes in there. And remember to come up with a spokesperson. And then you may find people popping in. So I'm going to open the rooms now and from the breakout rooms. So let's start with June and Noah to explain this idea of the role of urea in helping you make a gradient in the medulla and why that's needed. So do you guys have a spokesperson?
Yeah, I can go ahead and explain. Do you just want me to explain urea recycling or answer question 9?
Both. Yeah.
Okay, I'll start with question 9, just to give me a
Okay.
foothold. What does urea do to osmolarity at the deepest part of the medulla when ADH is present? So basically the thick segment of the ascending loop and most of the collecting duct is impermeable to urea, and which means that the deepest part of the medulla and the deepest part of the collecting duct, there's actually urea carriers, and these pretty much perform facilitated diffusion of urea into the medulla, which makes the medulla have a very high osmolarity. And basically when ADH is present, you do 2 things in the collecting duct. You insert aquaporins and you insert more urea channels or carriers. And essentially you have more flow of water into the medulla, which allows for higher reabsorption of water. And you have more urea come out and make the osmolarity of the medulla higher. And because of this, the thin segment of the ascending loop is actually permeable to urea. And since you got rid of most of the urea, at least like 50%, if I remember correctly, in the PCT, at the bottom of the nephron loop, there's not that much urea. So it's going to follow a concentration gradient from the medulla into the thin segment. And this is actually going to bring up the osmolarity in the nephron loop. It's pretty much going to go back up from 1200 to 700. And then once it gets into the thick segment, all of the pumps can pump out the salts. So once you get to the DCT, you pretty much have low volume of water and pretty much a high concentration of urea. And that's going to go from the DCT back into the collecting duct. And it's sort of like a circle that just continually happens with just some urea escaping, but some urea will go back into the medulla and then repeat this process over and over again.
Great. Thank you. That was really good. I thought I'd share the screen and kind of walk through what he was was just saying. Let me get rid of this breakout room picture. And again, I can't reiterate enough. I think why isn't it doing it? Nice. Okay. Let's see if oh, there we go. Those two little videos I think will help. They're not very long that Noah found. And I think they did a really good job of this. So on this figure, I know it's not as nice artwork as you have in those videos. He's really good at writing nice and neatly. You have this is the glomerulus and the PCT. Remember, this is all in the cortex, and that the osmolarity in the interstitial fluid of the cortex, the blood of the cortex, and in the filtrate is somewhere 285 to 300. I just wrote 300 to be simplified here. Now, as it comes out of the PCT and enters into the descending loop of Henle, what you're trying to do is get water to leave. So water does leave here, going into the vasa recta, being picked up by the vasa recta. And as water leaves, what it leaves behind is salts, sodium chloride being the primary ones, and urea. So you are removing water, that's causing a concentration effect, so that by the time you get to the deepest part of the medulla, the filtrate, which continues to lose water all along here, gets more and more concentrated. And about half of that solute that is in the filtrate at this point, down at the deepest part, about half of it's sodium chloride and half of it's urea. So this part, as Noah said, is largely impermeable to urea. But as you ascend through the thin segment of the collecting duct, permeability to urea will go up. Now, when we moved urea earlier, it was paracellularly. But when you're moving it here and in the distal part of the collecting duct, it's moving through carriers, like Noah said. So you can regulate the number of those carriers and therefore change the permeability of particularly the collecting duct here. So now why you want to do this is as you're going up in the, let me change colors here, as you're going up, what you're going to be doing is moving, remember, sodium, potassium, and chloride. I didn't put potassium down here, but it's certainly there. It's just not in high amounts. You're going to be pumping that out frantically, right? So that you can make the osmolarity in the upper part of the medulla, the part closer to the cortex, is being largely influenced by this high level of sodium chloride and potassium that's in the interstitial space, and that's responsible for pulling the water out up here. But down here, you don't have those pumps. You have a high amount of urea. So let's make that a different color. The concentration of urea is really high down here. And it is allowed to go into the filtrate in this thin segment. So this is permeable to urea. As Noah said, if ADH is present, the permeability of this part, only this part of the collecting duct, is permeable to urea through those carriers. And those carriers are inserted when ADH is present. I actually don't know how permeable it is when there's no ADH, but it's probably kind of a moot point because you release ADH enough that urea concentration will go higher. Now remember, this is passive, so it can only go from high to low concentration. That's what all that urea can do. Well, how does urea concentrate? Well, when you think about it, if half of it here is urea, half of it's sodium chloride, as it goes up and you remove the sodium chloride, most of what is making the osmolarity is due to urea. So you have an increasing urea concentration here. Now, why? Because you're removing the other solute. Yes, the osmolarity is dropping here, but most of this osmolarity is due to urea. not so as much to sodium, potassium, or chloride because you've pumped those out. So it looks like this, you would say, oh, that looks dilute. But remember, the volume has dropped substantially from over here, too. That volume dropped because water had been removed. Now you're removing the salts, and so what you're leaving is a very high concentration of urea. Now, when ADH is present, and this I think is maybe some of the next group's topics, when ADH is present, let me change this again, water is going to be coming out along the collecting duct. So this is all with ADH. You have water coming out all along the collecting duct and including down here in the distal part. So up here, that water is following the high levels of sodium, potassium, and chloride that are in this interstitial fluid. So water is following its osmotic gradient passively. It's coming out. Well, if you remove water, the osmolarity, I probably should change it again, is going to start to increase, right? So we're going to be increasing to, in theory, as much as 1200 down here. And, we're starting at 100 here. So if we move water and most of what's in there is urea, that means that the concentration of urea is going to even build higher. So let me switch to the green color again. So you have water removed and you're increasing urea concentration. So that as water comes out, the filtrate becomes more and more concentrated in urea. Now, it's not permeable up here in the part of the collecting duct that is sensitive to ADH, but not by inserting urea transporters. Instead, the largest amount of water that's coming out here is following the sodium, potassium, and chloride high concentration in the upper part of the medulla. That allows you to make this high concentration of urea. You don't let it leave, but you let water leave. As that urea gets so high up here, if it stayed really high, then it would keep the water from leaving down here. Water wouldn't be able to exit because you have a high amount of urea. So by allowing urea to also go out, you allow water to follow. So as urea exits here, it's following its concentration gradient. You've concentrated it, so it's now higher than anything that's out here. And it comes out, and that allows water to follow, which means that you are able to concentrate your urine even more. So down here in the deepest part of the collecting duct, you have this high concentration of urea. It's not as high as when ADH is present in the filtrate here because it's still going to be able to go down. Now, if that just stayed there, eventually you wouldn't move urea out at all, right? Because urea is passively moving. So you wouldn't be able to move more urea if you just left urea out here at a really high concentration and increasing concentration. So urea then is brought back in to the thin segment, and it means that it gets, as the guy mentioned, it gets recycled. So you end up using that urea down here at the distal end to allow you to pull water out. And you don't want it to build up too much, so you allow this thin segment to be permeable to the urea, the thin segment of the ascending loop, so it can re-enter and then contribute later again to the ability for water to be removed. This is not well explained in the book, so that's why I encourage you to think about this, be able to explain it separately from what's going on in just osmolarity. And what I mean by that is not just look at these as numbers, because we tend to fall into the trap of saying if it's 300 here and it's well, 300 somewhere. Let's say it's 300 right here. I'm just randomly picking a place. Let's say right here it's 300, that those are the same 300. In other words, this is the same solute when it's not. Over here, there's a lot of sodium, potassium, and chloride that later gets removed here and sent back into the vasa recta. So that the main solute here is urea, and the main solutes here, well, a main part of the solutes here, as Noah said, are the sodium, potassium, and chloride that are later pulled out in the thick segment of the ascending loop. So if you have questions about that, I know it's something you may want to kind of just sit and digest and think about and watch that video. It may be a couple times. There's 2 videos from the same person. It's just a continuation of his videos. So we finished 9 and 10. So the next group is going to look at a new part of the nephron, and that is going to be this part, the DCT, and discuss what hormones act on the DCT and what are their effects. So remember, the filtrate's coming in to the DCT, and it does have electrolytes besides the urea. It's not all urea, just the majority. And we're going to see what hormones act here. And this will be the beginning of our discussion of electrolyte balance that we'll go into more in the next chapter. So who is the spokesperson for group A? You got a spokesperson? So I'll just pick one if you don't speak up.
Professor, quick question. I never answered question 10.
I think you did. You said, what does urea do to the osmolarity in the deepest part of ADH?
That was question 9 for me, question 10 for me was what hormones act on the DCT, unless my sheet is wrong.
Oh dear, your sheet numbering and my sheet numbering are different?
Yeah, maybe I messed up somewhere, so it's okay then.
No, okay. Maybe that's the confusion here for the group A people. So I have for the question 11, what hormones act on the DCT, and for each hormone state the effect of that hormone on the permeability of the ion. And then 12 becomes what hormones act on the collecting duct, same sort of question. So let me just see somebody here. Who's in A is Angela, Jeanette, or Michelle. Who wants to speak up?
Yeah, those were the questions that we answered.
Which ones did you answer, Michelle?
11, 12, and 13. So which hormones act on the collecting duct? What are the intercalated type A and B cells? And low level of ADH effect on urine osmolarity.
Okay, so for you guys, it looks like 10 is the DCT one. Do you want to do it again, Noah, then if you...
Sure, it's a quick one. What we wrote is that there's 3 hormones, aldosterone, PTH, and AMP. Aldosterone will increase sodium absorption and increase potassium secretion, and it allows for chloride to be reabsorbed to allow for electrical gradient balance. PTH will allow for calcium absorption increase, and it increases phosphate secretion, and AMP antagonizes aldosterone and just inhibits its effects.
Okay, and sodium. Now these guys, oops, this is with aldosterone. Sorry, I'm being kind of messy here. And then we'll put a red one for AMP. All right, so that is true. These are linked ions, though. So the carrier for aldosterone is going to involve the sodium-potassium pump. So you're going to increase sodium reabsorption. And with aldosterone, that's going to go into the blood, but what's going to go into the filtrate is going to be potassium. So just like anywhere in the body where we've talked about the sodium potassium pump, sodium goes into the blood, but in this case, potassium goes into the filtrate. So you lower your potassium, you raise your sodium. Again, we did this in 103A without talking about exactly where this happens. So this occurs because aldosterone causes vesicles that insert the sodium potassium pumps into the membrane. And then when there is an aldosterone, they go back into vesicles inside the cell and become less permeable and don't pump. This is a secondary active. These guys are, primary active transporters that they're inserting. I hope you can see that into it. So what happens is ANP, when it's released, it blocks that effect. It blocks the aldosterone might be present, but it doesn't allow the insertion of the sodium potassium pumps into the membrane. Now with PTH, maybe I should make a little more room because this is a little newer with PTH. Let's go back to I use that weird green, so I'll use this green. With PTH, what happens is you increase calcium, just like Noah said, back to the blood. So that goes to the blood. And at the same time, is there a question? At the same time, this is actually a co-transporter with sodium, but I'm not going to worry about that at the moment, so that you actually are bringing some sodium in as well. It's a symporter with sodium. But what it also does, which is more important in a way, is it causes a decrease in phosphates. So it uses two separate pumps. It's not like the antiporter of the sodium and potassium. This goes to the filtrate and you lose phosphate. Now, why I want to bring this up is we will be talking about electrolyte balance, and that will be one of the electrolytes you're going to be looking at. But it's also, remember the other effect of PTH. PTH goes to the bone and breaks down calcium phosphates and causes those osteoclasts to transport not just the calcium, but also the phosphate into your body fluids, into your extracellular fluids. Now, normally, a phosphate should be at very low levels in the extracellular fluids. And calcium, you're trying to regulate calcium into that very narrow range that you might remember from last semester of 9 to 11 milligrams per deciliter for calcium in extracellular fluids. And that you need critically to keep within that range. So when you're trying to bring up the calcium with PTH by breaking down the bone, so put this should be very low. I won't give you a number. You end up raising your phosphate levels. So the kidney is helping you not just reclaim the calcium that might be lost in the filtrate by the way the glomerulus worked, but it also helps you secrete the phosphate, which comes up higher than it should be when the PTH breaks down the bone marrow. Is that clear? This is a lot of what we're going into in the next chapter, and we're not going to go much beyond that. So it's kind of a repetition, but I wanted to sort of stick it in here, and that will let us go a little faster when we get to this electrolyte balance. Okay, all right, great. So now we can go on to the group with Jeanette, Michelle, and Angela. Are you the spokesperson for that group? So this was question 10. Were you the spokesperson for that group, Michelle, or is there somebody
No,
else?
it was me, Dr. K.
Okay.
So we're answering what hormones act on the collecting duct. And we said that at the distal end of the collecting duct, the antidiuretic hormone allows urea to move out of the filtrate via facilitated diffusion. So when ADH is present, The distal collecting duct allows the area to follow its concentration gradient out into the deepest part of the medulla's interstitial fluid. And it also increases the rate of sodium potassium chloride pumping. And this
Hold on. Oh, I see. Okay. Yes. Okay. I know where you're going. All right. Never mind. I better write that. Yeah. I thought you're going to do aldosterone's thing. Okay.
And this increases the osmolarity of the interstitial fluid. And so water leaves the collecting duct to be reabsorbed into the vasa recta.
Okay. Great. So ADH there and there. And then what else does it do that you just said at the end?
So water is able to leave the collecting duct to be reabsorbed into the vasa recta.
Right. Great. So all along here, it's making water go out, right? Including down here. Sorry, it's a bit messy. I'm trying to keep up. Increases aquaporins. So remember, water is also moving passively along its osmotic gradient. And kind of like with the aldosterone, it can insert these aquaporins rapidly, or it can pull them out of the membrane into vesicles. So that allows the DCT to quickly respond when you need to concentrate your urine. Okay, so that's a bit of a rehash from where we were before. And did you cover aldosterone too?
No, we didn't do aldosterone.
So like the DCT, it's the same idea as the DCT. Aldosterone is going to insert sodium-potassium pumps that allow you to bring in the sodium, and that'll be picked up in the vasa recta. So remember, a lot of sodium's gone out, but you have a bit. So that's going to act both on the DCT and on the collecting duct. And meanwhile, it's going to cause secretion of potassium. These are employing the antiporter, the sodium potassium pump. Oops, this is potassium. So potassium is going into the filtrate and sodium is going into the blood. So if you need to, and maybe I should have made that red, if you need to, you can block this with A and P. So that would block this effect, and you would no longer have these guys working. They wouldn't be inserted, and therefore you could lower your sodium level. And if you lower your sodium level, less water will follow, and therefore you will increase your blood volume, decrease your blood volume. Less water would follow, the water would stay in the filtrate, you would urinate it out, and this would lower your blood pressure. Okay, I kind of went through that rapidly. We will be coming back to this, but probably without the picture for whatever that picture is worth. Are there any questions on this area? So you guys did 13 as well, right? Then, or well, what you called 12 then. My numbering must be off.
Yeah, but our
roles of the type A and B cells.
It was pretty short. We didn't find anything in the book that elaborated, but it's just important for the acid-base balance. So the type A and B cells will secrete protons and bicarbonate. And that's all we got.
Okay. So back when we talked about the collecting duct, most of the cells are the principal cells. And these are the ones that principal cells. These are the one that ADH, AMP, and aldosterone act on. And these are the majority of the cells. Most cells are principal cells. But embedded between them, and that's how they get their name, intercalated. Intercalated means, randomly scattered amongst all these principal cells are type A cells. And as she said, these allow you to secrete H+. And you do that not by a hormonal regulation. You're going to do it by a pH. So what stimulates this is low pH. So we will come back to this. It's the very last topic before the next exam. It's a challenging one. So I want to make sure we give more time to this than maybe the water balance, which is a bit more of a review, and the salt balance that are a bit review from 103A. So we have enough time to really look at this. Now, the type A cells are secreting H+, and these are the ones that are going to be mostly active, since, as it turns out, almost all of the waste products that your body makes when it does metabolism are acidic. There are a few conditions that can occur where your pH becomes more alkaline, and that's what the type B cells are doing. They are going to secrete bicarbonate, and they are stimulated by high pH. Now, that's not going to happen as much, so the book isn't going to say a whole lot about B cells. We're not going to go into their mechanism, but you can almost think of them as a turned upside down type A cell. This is rarer that you would have these cells be active because it's not common to become, to have alkalosis, to become high in pH. But if you are, then these cells can step in. So these guys regulate pH of your blood, and they're stimulated by abnormal pH levels. Okay, let's go on to the next group. And I think these questions, they're a little bit related to each other. 14 is related to something we just covered. It's kind of reiterating it. But then 15 and 16 start to look at what is in the urine. So who is the spokesperson for group C? No, it's group B. That is the one that says room 3 with Augustine, Ileana, Kaylin, and Prabdeep. Who's the talker here?
Dr. Kate, we actually answered that one if you want us to answer that.
Okay, so my numbering being off again, you did it as well. Which one did you do? The 14 on the effective low ADH level?
Yeah, the low levels.
Okay. All right. Why don't we do that? It is related to what you just did.
Yeah, it's pretty short answer too. We just said that if we're overhydrated, we don't have ADH, and the low blood osmolarity would mean that ADH. Wait, sorry. Oh, so low ADH would mean that we the distal convoluted tubule and the collecting duct would lack aquaporins. So we wouldn't be able to reabsorb the water, and we'd have a large volume of urine.
Yeah, large volume, low osmolarity, right? So remember how the urine came out at 100 way over here, wherever I did it? Here it is. It's at 100. If you don't have the ADH to insert all these guys, water doesn't leave. And in the case of low AD, you can make it as dilute. Where did I write it here? As dilute as 100 milliosmol, which means a third of your blood osmolarity. That means you can secrete a lot of excess water that you're intaking. And that's what I find most students are in the state of when we are in person in the lab and do a urinalysis. You'll find that you have very low urine osmolarity. Now, we don't actually measure osmolarity, as you'll learn today in lab. You measure something called specific gravity. So we'll talk more about that in the lab. Okay, all right, now what's in the urine? I think that's the next group. And that's the one that was called room 3 with Augustine, Ileana, Kaylin, and Prabdeep. And who's talking for that group?
It's going to be me, Professor Pradeep.
Okay, great.
Okay, so for number 14, what we put down is the four components of urine are urea, uric acid, urochrome, and creatine.
And creatine. Okay, so that's, I'm going to stick these together. Okay, and I thought maybe I've rewritten this question, but I have 6 normal components, so I'm just going to add some water and some salts. That's 5. So I hope my questions are not looking all that much different than yours. So the reason I put uric acid up here with urea is that you don't make much uric acid, but there is some in your urine. The majority of the nitrogenous wastes are urea. Now, why you want to do that is urea is fairly chemically inert. It's not a highly reactive molecule, so it doesn't damage your tissues as much as uric acid would. I mean, clearly this is an acid, so it's going to be chemically a little more active. So most of this is converted into urea. And when we get to the liver, and we'll see how the liver contributes to that later in the course. So what is this stuff urochrome? That's not asked in the question, but this came from eventually the breakdown of red blood cells. So the heme group becomes bilirubin, and then the kidney converts that bilirubin into urochrome. Creatinine comes from the creatine phosphate of muscle tissues and are the primary, but also the liver. So this is from creatine phosphate. And it's often used in urinalysis as kind of a hallmark if you have abnormal levels of creatine, especially if it's high levels of creatine. It can be a hallmark for liver damage. And then, of course, waters and salt, because you have to urinate something, and so there has to be some volume to it. So you need some water in there. Okay, great. Now, the other thing on here that could be in there is kind of in the topic of the next question, which could be toxins and drugs. So are you still the spokesperson for the question about renal clearance, and what is renal clearance?
I will be talking about renal clearance.
Okay.
Renal clearance basically measures a substance's disappearance from the plasma per minute. And inulin is the standard because it's neither reabsorbed or secreted from the filtrate at 125 milligrams per milliliter. And this is used clinically for determining how quickly drugs leave the body. So if a substance's renal clearance exceeds inulin, then that means it's being secreted. And if a substance's renal clearance is less than inulin's, then it is being reabsorbed from the filtrate.
Great. Perfect. So inulin is used because the kidney ignores it. It doesn't reabsorb it. It doesn't secrete it. It just gets filtered at whatever rate. So it's kind of like watching, something get pushed through the glomerulus. It just stays in the filter. Filtrate at whatever rate it's pushed. Sorry, it crashed. Zoom crashed for me. I don't know if it crashed for you. So I'm hoping do we lose everything? Yeah.
Dr. K, can you repeat what you said about
Yeah.
the insulin when it gets
I'll go back to know the definition. Let me share my screen again. It's a bit messy. I'm having a messy writing day, like some people have messy hair days. Renal clearance, I would know as a definition. And the reason I put it in here is because it is our kidneys and our liver are the main way in which drugs are removed from our bodies, clearly toxins as well. But drugs being removed this way, you need to know how long a drug stays at a certain level to be therapeutically effective. And some drugs have a certain amount of what we call side effects, but they could be toxicities too. So you don't want to keep them at too high a level too long. So what is often comes up as you go into the health sciences is that you're worrying about how frequently someone takes, you're trying to keep them taking their medications at a certain rate. And a lot of that is determined by looking at renal clearance. How quickly, well, assuming the drug is cleared by the kidney and not just metabolized by the liver, how quickly does it show up in the urine? Because that gives you an idea of how long you can keep that medication in the person and not have it be secreted out. And therefore, how frequently do they have to take their doses? So know that as a definition. How are we doing on time here? Okay. So let's move on to finally urinating. And the group that I called C on my page has the questions 17, 18, and 19, which are mine are the arrangement of smooth muscle and the internal versus the external. And that may be how the
yeah, we only show 18, so we did 17 and 18.
Yes, all right. So I think what I did is I dropped that question 17 out of the version you have, and that's why the numbering is off with my Word automatic numbering system. So you're just doing the two questions about holding and voiding urine, is that right, Tina?
Yeah.
Okay, that's what I did, and that's how my numbering got all off. All right, let's start with holding, and why don't you let us know what parts of the body are involved and what kind of reflexes occur.
Well, the stretch receptors in the detressor muscles, excitation of the sympathetic innervation signals the detressor muscle to relax, which enables the storage of urine. The parasympathetic plexus is inhibited to allow the bladder to fill. And then the pontine holding center receives the conscious sensation of the bladder filling, the conscious sensations of the bladder filling, and those are relayed to the brain from the bladder via the pontine. It functions as an on-off switch that is signaled by the stretch receptors of the detressor muscle and is regulated by inhibitory and excitatory signals from the brain. Excitatory signals cause the bladder to store the urine. Sympathetic innervation of the bladder regulates the storage of or holding of urine, and the somatic innervation of the bladder allows us to consciously and voluntarily control the closing of the urethral sphincter and also the opening of the sphincter.
Okay. So I'm trying to make this as a figure.
Yeah.
And external sphincter. All right, so you have stretch receptors in the detrusor, as she said, and they're going to send signals through the sensory pathways, coming from your up your spinal cord to eventually your pons, where there's a nuclei called the holding center, which is a great name because it's telling you exactly what it's going to do. It's going to cause you to hold your urine which hopefully you're doing right now. And it's going to do that by reinforcing what's going on at rest. So at rest, your sympathetic is exceeding your parasympathetic. That's kind of the reverse of what we normally think about with parasympathetic, sympathetic systems, but it is what happens here. The sympathetic is inhibitory on the detrusor, so maybe I'll do that in red. So this is going to inhibit the detrusor, or all the muscles, bladder muscles. It's going to cause them to relax. And it's going to, as she said, it's going to excite the internal sphincter. This is part of the very top of the urethra. You have a smooth muscle, again, visceral smooth muscle that you excite, and the sphincter closes when it's excited. The parasympathetic would allow increased activity in the bladder muscles, and they start twitching, and your internal sphincter might open a bit. So you inhibit all that stuff. And at rest, you also have your external sphincter kind of chronically excited by your somatic nervous system. And you're going to increase that when you're trying to hold your urine. So external sphincter is skeletal muscle, and internal sphincter is smooth muscle, and it's visceral smooth muscle. So you can do this once you're not an infant anymore. So somewhere, you get better and better at it as you go from infancy into young childhood. So what do you start potty training kids about? Maybe 2 years old, something like that. Before that, you're just fighting what they can't do. They can't do it yet. Now, why they can't do it, as I mentioned in the lectures, is because the inferior part of your spinal cord isn't fully developed at birth. So if you were an infant, or if you are now an adult and you went into the bathroom and you decided, good time to go, tell us about the reflexes that allow voiding urine.
Stretch receptors of the detrusor muscle send signals via visceral afferent fibers to the sacral region of the spinal cord. The visceral afferent impulses are relayed by interneurons to excite the parasympathetic neurons and inhibit the sympathetic neurons. This triggers the micturition reflex and causes the detrusor muscle to contract and push urine out of the bladder and into the urethra.
And what happens to the sphincters?
The internal, they relax.
Right. Increased bladder contractions, so they're excitatory here.
Right.
And then they're inhibitory on the internal sphincter. And this is all suppressed. And you can't do this just through your spinal reflex. Once you are an adult, you also have your external sphincter. So I'm going to make that a different color. So in adults, you also have to add a inhibition of your external sphincter. And you might say, well, what about in the infant? Well, the external sphincter isn't really working at all. So it's always relaxed. So this will relax and open. But in an infant, they're just working on their internal sphincter to relax and open. Okay, so when you go into the bathroom, as she said, you do it because your detrusor gets a certain amount of stretch on it that causes excitation to go through a spinal reflex. But if you're an adult, you can inhibit this reflex through your holding center. This happens, I think if I remember rightly, Dina, you can correct me, at about every additional 100 mLs, was it?
It was, say that again.
Was it like at each 100 ml increase in the bladder volume will cause a little excitation from these stretch?
Kaylin in chat is saying 200. I don't have that.
Okay. All right, maybe it was 200 and I've forgotten. The reason I wanted to bring that up is, remember, these are our sensory cells, and at a certain amount of stretch, if it's chronic, they're going to eventually become adapted. So if you decide to hold, like right now, let's say you're deciding to hold your urine, you had that coffee in the morning, and it's stretched and they're starting to get excited. You see little contractions happen in your bladder because of this reflex, but you are suppressing it. And after a while, those stretch receptors will go, okay, we're now at a new set point, and they'll quiet down and you won't feel like you have to pee. This will go on until you get about a liter of urine, and then it all heck breaks loose and you can't override the reflex anymore. So there's a limit of how much urine you are able to, sorry, my dog is talking. You're able to hold in your bladder without this holding reflex being overridden. All right, so I think this is a nice figure in the book. I kind of wish they had done two figures, one for holding and one for voiding, so that you could see that. And so this little bit of a skeleton, I'm hoping, will help you. All right, so the last group today, which we should be able to go into, sort of backs up and goes back into the kidney and into what are the normal conditions of body fluids in your body. And we'll start with ions and body fluid normal distributions, and then see how we can move some of these things. And if we have a chance, I'll start into some of the negative feedback loop, and then we can move some of that. We'll see how far we get. So this last group, and I just had this fall asleep again. The last group, and I don't have who was in it, but I could in a minute. Is there a spokesperson for the first four questions of lecture? Okay, great.
Yeah, so I'm doing question 1, and that's what percentage of body fluids are each of the following?
Okay.
So for ICF, we have that it would be 40%.
Sorry, I can't get this thing to share. 40 ICF.
Yeah.
It's not working very well again. All right, I'll just do it on if I can on here. ICF is what?
40%.
40% of your body fluids. Water, I guess, fluid in your body. And what's the other part?
So for ECF, it would be 20%, but of that, it's split up into plasma, which is blood, and then interstitial fluid. And so plasma would be 20% of ECF, and
And I have a little different numbers. This is 60%, right? Did I type it wrong? I have this is 60%. This is 40%.
Oh, yeah. I think I read the wrong thing. Sorry.
Okay. With 20% being, I think you were heading on the right track here then. Oops, 20% being interstitial
plasma.
fluid. Right. Okay. Sorry, I got it. 255%. Got better than you. Here we go. And the rest being what?
The rest will be plasma.
Blood plasma, right?
Yeah.
Okay, good. Now that's a little bit rough because you do have other body fluids that are not included. So we're rounding up to an even numbers here. What other things are extracellular fluids would be like the cerebrospinal fluid, the serous fluid in your peritoneal cavities and your pleural cavity, pericardial cavity, synovial fluid, and of course lymph. So they aren't, maybe it's even best to put this as plasma and lymph because that's probably the largest of all of them. And you even have fluid in like your eyeballs, right? You have that fluid. Okay, so do you have to memorize all these numbers? No, not necessarily, but understand that most of the fluid in your body is actually inside your cells. And we tend to think of fluid when we think of fluids in the body as mostly blood. We tend to think of it when really it only constitutes blood plus plasma, really somewhere around 20% of the fluid in your body. Now you need to keep this in balance, and the book uses, and I think in 103A, at least I used, the idea of compartments, that water can, with a rare exception, move between these compartments, between intracellular and extracellular fluid osmotically. The exceptions are like the descending loop of Henle, or the ascending loop of Henle, that doesn't have any aquaporins. But most of the cells of your body have many aquaporins and allow water to move. So you need to maintain an isoosmotic relationship between the fluid in your cells and the fluid in the especially the interstitial fluid right outside the cells. That depends on salts. So this is a bar graph in the book that you should know most of. I X'd out a few things that you don't have to worry about. So let's talk about, let's put extracellular fluid here and maybe intercellular fluid here. Where are these salt concentrations? High and low. Is this you? This is the next question. Is this, forget who was talking, I'm sorry. Who's talking for?
Yeah, it was me, Minri. For number 2, we know like where each one is high and which one is low, but we're not sure like why. So I can tell you like the first part though.
Okay.
So sodium will be high in the ECF, and potassium will be low there, but it's the opposite in interstitial fluid.
Okay, sorry.
Okay.
I did this wrong. High potassium, low potassium. And there is a particular reason that we drummed into you in, let me see if my pen will work now on this. It stopped talking easily to it. Can I write on this now? Yeah, it stopped talking to it easily. Okay. Oh, it's just slow. The main pump here, sorry, I scribbled all over it now, is the sodium potassium pump, right?
Mm-hmm.
Cells have sodium potassium pumps. Yeah, everything's just going really slow. After it crashed, it just isn't what it once was. So all cells have sodium potassium pumps, and they're kicking out 3 sodiums, which is making sodium high here and sodium low there. And they are pulling in 2 potassiums and kicking out 2 potassium. Pulling in 2 potassiums from here, making it high inside. Why are they doing this? What are they trying to maintain? Anyone remember from 103A? The membrane potential? Do you remember this? I don't know, maybe it's all so long ago, but hopefully Dr. Sundar drummed it in as well, I'm sure he did, that in order to maintain a normal minus 70, well, it varies from from different tissues, but in excitable membranes, around minus 70 millivolts, you need to maintain the ratios of sodium and potassium. The other reason you need to do this is osmotic. Water is going to follow sodium. So if the cell membrane were very permeable to sodium, which it's not, it's pretty impermeable to sodium, then as sodium, being that it is high out here, as it tries to go inside the cell, water would follow and the cell would start being osmotically out of balance. Okay, did you do some other salts, Manreet? How about calcium?
Yeah, for calcium, we have that there's no ionic calcium in the interstitial fluid.
I'm just going to say small outside and none inside. Now, why that's important, why it's none, is that any calcium that does come in gets sequestered. So you can go back to remembering muscles where it's put in the sarcoplasmic reticulum. But in any cell, it gets sequestered into the smooth endoplasmic reticulum, or it gets chelated. Chelation means when you bind calcium to another molecule. So now it's no longer ionic calcium. You don't count it as calcium. So that way, whenever you open a channel for calcium, like in the cardiac muscle where we open the channel to add to the membrane potential or to cause the autorhythmic membrane potential, it will definitely go in, even though its concentration outside is not all that high, as you can see if you look at the bar graph. Okay, so what other ions? Let's see what I have else on my list. What about chloride?
Chloride, it will be high in extracellular fluid.
Okay, and low in chloride. So this one is balancing the sodium. This is a anion, you need a cation to balance it. So, sorry, this is the cation and the anion. And in the case of extracellular fluid, it's bichloride. What is the negative ion that's high in intracellular fluid? It's going to balance this potassium. Anyone? No? Well, phosphates are 1. What is that, 4 minus? Phosphate is 1. Phosphate should be low out here, as I mentioned a minute ago, because of, we were talking about PTH's effect, that you want to get rid of that. Sorry, that's supposed to be a phosphate. You want to get rid of that phosphate. So it's higher inside the cell, and it's still not high, but it's higher. Now, why you don't need quite as much is because this guy is also balanced by negative charges on DNA, RNA, and proteins. So outside in the extracellular fluids, you don't have a lot of those items, so you don't need to balance it as much. Okay, I think that magnesium is on there and I got rid of it, so you don't need to worry about that on the chart. So we're just about out of time, but a question, let's just do question 3 if we can, because question 4 kind of leads us into what moves these ions, and that's a bit of a mechanism. So generally, if it's a gas, how does that move across a membrane? What mechanism? Yep. And similarly, if you have lipids, they're going to move that way, right? So let me get rid of that. Okay. This is, again, all from 103A, so you should know these from that. But if you've forgotten, I would review this. Most nutrients move into or out of cell, well, into cells at least, by secondary active transport. I'm going to just hurry up to get through this part, so sorry if it was you guys. Secondary active. And they can also use, to leave a cell, they can use facilitated diffusion. So go back to thinking of the PCT and how sodium pulled the glucose or other nutrients from the filtrate into the PCT cell But when it had to leave the PCT cell, there was so much of it, it was high concentration now, it could do that by facilitated diffusion. So those are the two ways in which nutrients tend to cross membranes. How about water? Water isn't using simple diffusion, it's using, and in particular, aquaporins. Again, very rich in most cells. And what else is on, on our list? Ions.